Tonight's lecture is titled The Genes of Genesis. And somebody pointed out to me that there's quite a similarity there between genes and Genesis. One could actually read genes is. And that's quite an interesting observation. You see, God also says, I am. He is, so why can't genes is? Well, we're going to talk tonight about the philosophy behind natural selection and the science of evolution in general. And the founder of this modern theory is Charles Darwin, who accepted a position of ship's naturalist on the HMS Beagle and studied the diversity of organisms while this ship was traveling around the world, in particular to draw maps of South America and the like. Of course, he didn't actually come up with the original concepts. They have been around for a long, long time, even since the time of the Greeks. And Charles Darwin's grandfather was already a proponent of naturalistic ideas. He also had theological training, and Charles Darwin believed at the time that he was active on this ship, he believed at that stage that God had created immutable, unchangeable species. You see, the mindset at that time was such that people believed that everything that exists on the planet had been created individually by God. And of course, naturalistic science excludes that possibility. Now, when Charles Darwin was traveling on this Beagle, he came up with the idea of natural selection, and we'll talk about that in some detail in a moment. Basically, this is the concept. If you have 3 giraffes, for example, the ones with the longer necks would have a selective advantage when it comes to feeding on high branches. And therefore, they would be selected for and have more offspring and eventually replace those with shorter necks. That's basically what natural selection teaches. There is a counter-concept which was known as Lamarckism. Now, Lamarck taught that the animals tried to reach the higher branches and eventually the necks got longer. You see? So those are the 2 opposing concepts that existed at this time when Darwin formulated his theory. Of course, Darwinism is recognized as the scientifically logical theory, whereas Lamarckism is discredited these days. I always put it to my students this way: do birds fly because they have wings, or do they have wings because they fly? Which one is correct? Which one do you believe is correct? Well, obviously they fly because they have wings. That's why they fly. So, you first have to have the wings, and then you can fly. And if you have different wings, well, then one can fly better than the other one, and then natural selection can start operating. Birds did not wish to fly and therefore developed wings, because then all of us would have wings too, right? Because man has been wanting to fly forever. So science tells us that the object has to be there before selection can take place, and that is pretty logical. So Darwinism is superior to Lamarckism. Now, it's interesting that science readily recognizes this, but when push comes to shove, then often they fall back to Lamarckism. That's very interesting. Remember yesterday I gave a lecture on the evolution of man. And one of the theories that they have as to why man began walking upright is that he lived in a time when the grasslands took over, right? And so they couldn't see over the grass, and what did they do? They stood up on their hind legs to look over the grass. And this eventually led to the development of upright gait. Is that Darwinism or Lamarckism? That's Lamarckism. Isn't that interesting? So science shoots down Lamarckism, but uses it when it finds it useful. Actual fact, you should conjecture differently on that issue. Now, remember what I said. When Charles Darwin was traveling on this ship, the Beagle, and he developed all these ideas and took them back into society, he was actually quite a shy man about it. But fortunately for him, he had another man, you can see with a much sterner expression on the face, Huxley, who was then known as Darwin's bulldog. And he took up the challenge and championed Darwinism to the scientific world, and is credited with conquering for science in a debate with Bishop Wilberforce, which he didn't actually win on the grounds of his science, but won on the grounds of the ridicule and contempt placed upon the science by the bishop. So you see, the attitude was what determined the winner and not necessarily the message, which is quite sad. And it's a good warning to us to be careful how we approach things. Now, Charles Darwin was very interested when they came around the coast of South America, when they came to this group of islands known as the Galapagos Islands. And on these Galapagos Islands, he found some very interesting creatures, some of which we looked at last night, like these giant tortoises and those beautiful Galapagos marine iguanas and all of those interesting creatures, but amongst them also these finches. And these finches were unique. Some of them had large beaks, some of them had small beaks, some of them had much larger beaks, and these beaks were adapted to certain circumstances. So, for example, those with the larger beaks could crush hard seeds and open them up and reach inside. Those with the smaller beaks had to feed on different things, so some of them even developed the method of using tools to get hold of grubs and what have you inside the bark of plants. And so each one of these was slightly different. Now this was something very strange. You see, on the mainland, songbirds are credited with this variety. But here the finches showed this amazing variety. Now, if you imagine the time frame in which Darwin worked, genetics was unknown. Gregor Mendel had done his work, but it lay somewhere on a dust pile in some monastery. Nobody knew anything about genetics. So how inherited characters were transferred was unknown. And then there was this idea that God had created immutable, unchangeable species. And there was a scientist by the name of Linnaeus who took all the creatures that he knew and classified them according to what was to be known as the binomial nomenclature. So everything got two names. So, for example, we are Homo sapiens. We are Homo sapiens, two names. And each creature got its name, and it was considered to be a unique creation. And so the species numbers increased enormously until we today have millions of species around. Now, when Charles Darwin looked at these creatures, he thought about this and said, now, isn't it possible that we started out with one finch family some time ago on these islands, and that all of these evolved out of that original parent pair? Isn't that a logical assumption? And yes, it is a logical assumption. It is quite probable that this is a variation that developed there on these islands. But what Charles Darwin did then is he said, well, then God did not create immutable, unchangeable species. In fact, there is no such thing as a creation. So God was thrown out of the equation. Was it necessary to throw God out? That is the question that we will have to answer tonight. The fact of the matter is, it does seem as though whatever was there was not unchangeable. Well, science has come a long way since then, and we have a whole theory of how everything came into being. And let's take it from the beginning and see what science teaches today. So how did life begin? Well, science teaches that this earth originally was very different to what it is today. There was a volcanic activity. The atmosphere was totally different to what it is today. In fact, the atmosphere consisted of gases like methane, ammonia, hydrogen, water, these components. And a scientist by the name of Stanley Miller came along, and he simulated this atmosphere, passed some sparks through it, and then trapped whatever was formed at the bottom and analyzed it in a laboratory. And lo and behold, here were organic materials. And so he conjectured that originally on the planet, there could have been an organic soup with lots of organic molecules, which then fortuitously formed themselves over long periods of time into the molecules which are necessary for life, and then through processes eventually came together to form the first living organisms. Now that's all very nice and well, except that there are a number of problems with this. Firstly, the atmosphere would have to be one that excludes oxygen. Very important. So, the primitive Earth-reducing atmosphere is supposed to have had hydrogen, ammonia, carbon dioxide, methane, nitrogen, and water. And where did these come from? Well, they stated from volcanic gases, although volcanic gases are also associated with carbon monoxide, sulfur dioxide, so it doesn't really quite fit, but nevertheless, if it had, however, been like the present atmosphere, which contains oxygen, then the evolution of molecules would have been impossible, because oxygen burns up everything. Oxygen would destroy everything as it developed through the process of oxidation. So all scientists agree it would have to have been a reducing atmosphere. Now, what did Haldane do? He passed sparks through such an atmosphere and then trapped whatever was formed and analyzed it. If he had left it there in the atmosphere for another cycle, then everything that would have formed would have been destroyed by the next process. So he had to have a trap. Would there have been a trap somewhere on the planet before? Probably not. But there's another problem. And that is the various molecules of life that had to form needed very different circumstances. For example, if you look at the amino acids, you have different amino acids, and each one of these amino acids has this NH2 group, this amino group over here. And this NH2 group had to, of course, be derived from this atmosphere, and it comes from the ammonia. But now, if you have ammonia in the atmosphere, then sugars don't form. So here is a big problem. Plus, you need a very acid environment in order for these to form, and then you would have to have only the right ones to form. And that would not happen, because when you pass these sparks through it, you get left-handed ones, you get right-handed ones, and you get all kinds of amino acids where the amino group is attached to any one of these carbons along the chain, and not necessarily to that first carbon next to this group over here, which is the carboxyl group. And so, if you have just one wrong amino acid somewhere in a chain, everything is non-functional. So here's a major problem, that you needed the right circumstances, plus you needed mechanisms of selecting just those amino acids which are useful to life. One wrong one in there, and you would have a problem. Plus, even if you get the amino acids, how do you get them to come together and join together? You need enzymes for that. That means you need a protein to make a protein. So who was first, the protein or the protein that makes the protein? See, it's a bit of a problem. The next problem is, if you want to have the building blocks of our DNA, what our genes are formed of, then you need totally different circumstances. For these to form, you have to take all the nitrogen out of the atmosphere, change it into a cyanide solution, multiply that by 10, dissolve it in the ocean, and then you can get these molecules. But then you wouldn't get anything else. If you're going to make these sugars, again, as we spoke about earlier, that you need in the DNA molecule, for example, like the ribose, well, then you're not allowed to have ammonia. If you don't have ammonia, you don't have amino acids. So you would have to have many planets in order to create these things. No wonder science is saying it's not possible here, so maybe it happened in space. You have a theory there which is called panspermia. Now, this DNA molecule is another amazing story of life. Even if you get all the building blocks to form, how would you get it to form a string containing all the information that you need for life? How would you get it to do that? It has never, ever been demonstrated in a laboratory. It needs a very complex enzyme system that will form these high-energy bonds that are locked into this molecule. And the probability of these molecules forming is so remote so that we could say they are non-existent. So let's have a look at what the probability would be, for example, of a bomb exploding under a pile of wood, going boom, and then falling down from the sky and forming this perfect functional little house. What would the probability be? Well, the probability is very, very remote. Let's give it a very good probability, or a very poor one. In fact, it's even much worse than this. Let's say the probability is 1 in 10 to the power of 80. Now, what is that? That sounds like a very small figure. 10 to the power of 80 is what physics claims is the number of particles in the entire universe. Particles. That means not only atoms, but subparticles. Electrons, hadrons, quarks, neutrons, protons, in the entire universe. How many atoms in a pinhead? Billions and billions and billions and billions and billions and billions, right? So imagine how many atoms in the entire universe. It sounds mind-boggling, but it's only 10 to the power of 80. It's not a very big number when you look at it, but that is really a mind-boggling number, 10 with 80 zeros behind it. So that would be the probability. What would be the probability of a simple gene coming into existence by chance? Well, there are 3 nucleotides needed to code for 1 amino acid in a protein. And let's take a simple protein that has 100 amino acids. Your hemoglobin, for example, has 600 amino acids. So let's take a simple protein, 100 amino acids. You need 300 nucleotides in the right sequence. What's the probability of that happening by chance? Well, the probability is 10 to the power of 127. That is, unfortunately, boom! New York City has been established by a nuclear explosion. Do you believe that? You would have to have that kind of faith. Now, if we look at this DNA molecule, it is a masterly construction. It consists of these double helixes forming the framework of phosphorus molecules, then the bases in between, and in the sequence in which they occur, these bases, you have the information for everything that happens in the cell. Now, these genes we call the genotype. And when these genes are expressed, we call that the phenotype. All right, let me explain these terms. It's very important that we understand how science pictures the evolutionary events around the genetic system. The genotype consists of all the genes actually present in a zygote. That's the fertilized egg. So all the genes that I have, that's my genotype. But not all the genes that I have are expressed. So that which is expressed is my phenotype. So if you want to have a look at your phenotype, go and stand in front of a mirror. And you'll see it. That's your phenotype. Now there's a law in evolution which states the following. Natural selection can only operate at the level of the phenotype, never the level of the genotype. Have you got that? Let me repeat it. Natural selection can only operate at the level of the phenotype, never the level of the genotype. Why not? Well, let me illustrate with an example. Two people walk in wherever, in a game park. And let's say there are wild animals around in the game park. And the one person is long and lean, and the other person is short and plump. And now a predator comes from behind the bush and attacks them. A lion, a bear, whatever you would like it to be. And they both take off like greased lightning. Which one is probably going to end up being a meal? The short fat one, right? Okay. So short fat was the what? The phenotype. That's right. Short fat phenotype hasn't got a snowball's hope in hell against the lean mean phenotype. So by natural selection, short fat goes, long lean mean stays. Is that correct? That's how it works. The animal is not concerned with all the other information in your genes. It's just interested in how you look and how fast you run. Is that correct? That's it. So natural selection does not work at the level of the genotype. So what does work at the level of the genotype? How are genes changed over time? Not by natural selection, but by mutations. That's right. And how do mutations take place? They are random. They take place by chance. And once they are expressed in the phenotype, then they can be selected for. Does that make sense? Now remember, Charles Darwin didn't know anything about this. Let me simplify this for you so that everybody can understand this. Now, I know that I'm speaking to some who have biological training and some who have no biological training. So I'm trying here to target all of you. So if I shoot over your heads tonight, there will be plenty that you can take home with at the end of the day in spite of that. If I shoot too low for your liking, then please tolerate that because there are people here that would otherwise take nothing home with them. Is that fair? Good. So let's sum up. This is your genotype. That's the information in your genes, in your DNA. The phenotype is what you look like. That's what we said. Natural selection operates at the level of the phenotype. It doesn't operate at that level. That's where chance operates. Chance through mutations. Fine. Let's put that into another model. Here we have a book of instructions on how to build an aeroplane. Okay? Here's a book of instructions with detailed instructions on how to build an aeroplane. The book of instructions in our analogy is what? The genotype. The DNA is a book of instructions on how to build a human being. Isn't that correct? All right. And the phenotype is the actual aeroplane. Now, question. Who wrote the book? Well, who wrote the book? Obviously, some intelligent designer wrote the book in this case, right? Correct? But in our analogy, who wrote the book? Chance. Or as you guys said, chance. I can say it. Chance wrote the book, just so that you have another accent to contend with. All right, Chance wrote the book. That's incredible. With all the information as to how to build the airplane. Now, that's fascinating. That is really monstrous. That's terrific. Let's play this game out. I have a book. Here it is. And let's assume, for argument's sake, that this book has now come into existence by chance, mutations. And all the information for whatever it is we're going to build is in here. In this case, all the information for my aeroplane has now by chance appeared, and here it is in this book. This is great. So now I'm going to take this book, and I'm going to put it down over here on the floor. There it is. It's in my bookshelf. There it is. And now I'm going to wait for the aeroplane to appear. After all, I have all the information. It has come about by chance. There it is. And it's all there. Now, all that has to happen is the aeroplane must come. How long am I going to wait? How long am I going to wait? Well, come on. Forever. I'm going to wait forever and ever and ever. Is it enough for the information to come into existence by itself, yes or no? No. If I don't have a mechanism that will translate this information into the aeroplane, then my aeroplane will never appear. Is that correct or not? Okay. So my book by itself, even if it should come into existence by chance, is totally what? Useless. Thank you. Okay. Now that we've solved that problem, let's think about that. All right. So my book is really useless without a method of transcribing the information into the aeroplane. Now, what do I need to change the information into an aeroplane? You tell me. Well, I need some factory, don't I? And I need someone to intelligently interpret what is in that book so that I can get the aeroplane constructed. So I need some assembly line, and I need some workers. Or if I don't have workers, what do I need? I need tools and machines, robots, if you like, that will do it for me. Is that correct? So, question. Where or how did the design mechanisms get translated? How did the factory come into existence? And how did the robots come into existence that will eventually build my machine so that I can test fly it? According to my analogy, natural selection will only start operating when once what has happened? The aeroplane's there. So the whole mechanism to convert the information into the aeroplane, how did that have to come into existence? You tell me. You have two choices. Two choices. Because natural selection will only work once the aeroplane is there. Then I can only see, oh, it flies or it does not fly, right? So how did the mechanism come into existence? Give me the choices. The first one is chance. The second one is design. Design. That's your only possibility. Either it's chance or it is creative design. Those are the only two possibilities that we have. Okay, which one does science choose? Chance. Science chooses chance. Good. Now, when we read the DNA, this is a very complex situation. In fact, we don't even read the whole of a gene. We read only portions of it called introns and exons, and the exons are transported and translated and taken outside, and then there is a highly complex factory with robots that changes the information from the gene, in other words, reads it, and then translates it here into another form of messenger, which then passes out through the cell, combines with a structure we call a ribosome, which is an assembly line, a factory, and this factory then manufactures a protein, which in turn is manipulated and folded by another factory and transported, like with a railway system, if you like, to where it is needed and built into place. How did all of that come into existence? Well, we're still not with anything tangible. We're just building something tangible. So how did it come into existence? You tell me. You have two choices: chance or design. Because natural selection will only operate once the object is actually there. Wow, that's mind-boggling. You want to see what it looks like? Well, let's have a look. Here's an Illustria Media video, which is titled "Unlocking the Mysteries of Life, the Scientific Case for Intelligent Design." You can actually get it at the back. It is sold at the back, and you can have a look at an animation of what happens.
With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
It came about by how? Chance or design. That's your only option. There is no other. In actual fact, what you saw in this animation is highly, highly oversimplified. When you consider what happens in a cell as that DNA unfolds and the enzyme systems that are required to produce this molecule, it is mind-boggling. And when it passes through that nuclear pore, it passes through with such precision that nothing else may enter and leave while this huge molecule is passing from one side to the other. Everything is highly precise with recognizing symbols and all kinds of structures. And one mistake and you're a cabbage. And it came about by chance or design. Because until that protein is actually there, natural selection cannot say whether it will work or whether it won't. Now, any modification in the genes, in the genotype, to bring about any changes in the phenotype must also occur only through chance, through mutations. So if we take my book scenario, here's my book. If this book has instructions for an aeroplane, and I now want to change the instructions to get another aeroplane, then how must those changes take place in here? By chance. That's right. You've got it. By chance. Or by design. One of the two. So now you get basic types of mutations, point mutations, chromosome mutations. Point mutations involve a chain or change in only a single nucleotide pair. You can have base substitutions, additions, deletions, all of those. Now, we've already said that the DNA code is a triple code. So let's say you have adenine, guanine, thymine, guanine, cytosine, adenine, etc. All along the line, there are 4 nucleotides. And let's say you deleted 1. You deleted 1. Boom, you take it away. Then instead of having AGT, GAG, GCA, you would have AGT, AGG, CAT, TCA, GC, GGC, and the T. And that means that the entire message from the second one on would be changed. What does that mean? Let's make that simple. Let's use a triple letter code, the cat and the hat. And I'm going to delete the C, and then my sentence would read the at. That means the mutation would have made garbled junk out of the rest of the genetic information. Is that correct? So that is why mutations generally are harmful, incredibly harmful. And this is not a mutation. He's just there to look cute. But what intricate design features are built into this creature really boggle the mind. And is there such a thing as a mutation that is useful? Science will tell you yes. They will quote sickle cell anemia and say, well, that's a useful one. You see, if you have the sickness, sickle cell anemia, and you happen to live in a malaria country, then the malaria parasite cannot get into that sickle-shaped red blood cell, and the people don't get malaria so readily, whereas other people do. So there you go. It is positive. Oh, no, it's not positive. Someone with sickle cell anemia is pretty sick. He's pretty sick, so it's not a positive mutation at all. Just happens to be so that in the case of malaria, he'll be slightly less sick than the one that has malaria, but he's still sick. So there is no real positive mutation whatsoever. Now, imagine now the first little cell comes into existence, just by chance. Highly improbable. The molecules, well, they would not have come into existence by chance under the different circumstances. Even if they did, would they form life? Highly improbable. You'd have to imagine boom, little house, boom, little house, boom, little house, boom, little house, thousands of times over. And you'd really have to believe that because chance or design is your only option at that level. And then, let's say, fortuitously, besides all that, a cell appeared. Now you have one cell. Now, of how many cells do we consist of? Thousands. And they're all different. So now, how do you account a unicellular little creature through evolution eventually giving rise to a multicellular organism? Well, a chap by the name of Haeckel came along and said, German scientist, that's not so difficult. What happened is the following. One cell came, and when it divided, that's another kettle of fish. How did it divide? But let's leave it at that. It divided, and the cells stuck together. And eventually it formed the ball of cells, and eventually this structure, because the ball, when it moved along, how did it move along? How did it get the structures to move along? That's another kettle of fish. Let's leave that out. And let's say it developed a dent, and eventually this dent was like this, and eventually the dent went all the way through, and then you have the basic body plan of every single living organism that is multicellular. Like we, for example. We look like this with a dent all the way through. We have a tube going all the way through, right? The one opening is where? There, and the other one, you can imagine. So that's the basic body plan. Now, think about this. If a cell divides and it has genetic information and it sticks together, then both cells will have the same genetic information, right or wrong. And if it divides again, it'll still have the same genetic information. And if it divides and makes a ball, they'll all have the same genetic information. And no matter how many times you divide it, it'll have the same genetic information. Haeckel says, we can see in the evolution of animals that this happened because the embryos of the various animals look the same. Of course, they don't tell you that he really cheated in this process tremendously and was even reprimanded at the highest level because of this. And how do you account then for this amazing feat of a mutation changing cells? Now, I can assume that a mutation can change the shape of a cell into something else. Let's say that's possible, but that's not what we want. We want to develop different cells. So, for example, let's take two. A muscle cell and a nerve cell. Two very different cells. Did they exist in the beginning, yes or no? Let's say fortuitously a little blob of protoplasm started to go bloop, 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 bloop in the ocean. Would there have been a muscle cell and a nerve cell, yes or no? No, that would have been some later innovation, right? So how did the muscle cell and the nerve cell get its genetic information? You see, a muscle cell must have genes which say you are a muscle cell. And a nerve cell must have genes which say you are a nerve cell. Does that make sense? Okay. But none of these existed in the beginning. But let's say that the first cell looked something like this. That could account for this shape, but it couldn't account for the next shape. Where did that new shape come from? So what we actually need is we need a new gene which says, hello, you are now a nerve cell. Does that make sense? But that's not enough. If I had a nerve cell in my body and I had a muscle cell here in my arm, what would it help me if the two never talked to each other? Would it help me? No. Would it help me if I had muscle cells and nerve cells all jumbled up in my body, yes or no? Of course not. Where do I want my nerve cells? Well, I want them in the right place. For example, it would be very useful if you want to study to have them all up here, isn't that right? And if you wanted to pick something up, to do it up with a brain cell, pick it up with a brain cell would be pretty useless. So you want your muscle cells down here, right? Okay, so how do I tell the cell the genes for muscle must be switched on here and the genes for brain must be switched on over there? How do I tell my brain that or my muscle that? Well, I need a switch. I need a switch. If I have two lights, one over there and one over there, and I want that one on and that one off, or that one on and that one off, how many switches must I have at the back? Two. That's right. So now I need a switch because I've got two cells. But the original cells didn't have two kinds of cells, so they didn't need the switches. So now I have a question. Where does the extra gene come from? That's genotype. How did it come into existence? You have two choices. Chance or design. So, boom, little house. Where did the switch come from? In fact, you need two switches. Where did they come from? Boom, little house, boom, little house. That's where they came from. That's what you have to believe. There's no other choice whatsoever. And this highly complex mechanism for switching on genes and switching off genes had to come about by chance or design. But that's not enough. You need a gene which says you look like this. You need a gene which says you look like that. That's two genes. You need a gene which says you're switched on. That's another two genes. Those are the switches. That's not enough. You also need genes to control the physiology so that this one will function differently to that one. And you need genes controlling the embryology so that they will work together. Hello, where do those genes come from? It's genotype. Natural selection doesn't work there until this thing actually works. Boom, little house, boom, little house, boom, little house, boom, little house, boom, little house. For a week you have to say that. You need one heck of a lot of faith for that. All right, we have been discussing in this previous little section of how the genes actually came into existence. They could only have come into existence by chance, and then you have this high improbability of it actually happening. Now you have another problem, and that is the problem of irreducible complexity. Something that is so complex that has to be complete before it will function has to also come about by chance. For example, a wing to fly and a feather to hold the bird up has to come into existence and actually function as a feather before it can be tested as to whether it will work or whether it won't work. Isn't that correct? So these structures, where do they come from? These are questions that are really causing Darwinism quite a headache, as you can see over here. Here's a problem for you. Assume now you find a highly complicated biochemical pathway in a bird. Something very, very complicated. Let's say the Krebs cycle. For any student who has studied this, you know there are many, many reactions and many, many genes required to make the Krebs cycle work. And then you find the Krebs cycle in this reptile, and you find the Krebs cycle in that insect, and you find the Krebs cycle in that crab, and in that worm, and in that worm, and in this echinoderm, and in these sponges. Whoo! Now you find the Krebs cycle in all of them. What does that tell you? It tells you that they must all be related, and since a bird and a sponge are very, very far apart, that means that this highly complex system must have been there from the beginning. Wow. So if you take all the complex biochemical pathways that are shared by every living organism under the sun, that means they were all there from the beginning. Whew. Now we're thinking, boom, New York City. Boom, New York City. With all the elevators working and the doors having locks and the whole tutti with the sirens blowing the works. Everything coming about by chance. You really have to have a lot of faith for that. Let's have a look at selection. Selection eliminates deviant phenotypes and normalizes the population if it's stabilizing selection. Let's look at just one form of selection. What does it do? It eliminates deviant phenotypes. Okay. We have two aeroplanes. One flies well, one does not fly well. I will select one and scrap the other. That's what you do. That's selection. Let's take that to biology. Let's look at the peppered moth. Because the population possessed dark genes, peppered moth have been adapted to increasingly sooty industrial areas of England. That's the theory that they go. And then they show you this model where you have a light moth on a light background and a dark moth on a light background, or vice versa, a black moth on a black background and a white moth now on a black background. And they say it changed because of the suit. Actually, that's a lie. It didn't happen like that. It didn't happen like this at all. They stuck these onto dark and light backgrounds. This is not what really happened. They just supposed that this could happen. All right? But let's run along with their theory. It's a nice theory. And say, now, let's say the area was generally black. Which moth over here would have a greater chance of survival? Obviously, the white one. No, the black one. Why? Because the birds can't see it. So the bird comes and eats the white one. Doop, doop, doop. what eventually happens to the white one? It disappears. What do we call that in science? Extinction. Natural selection eliminates those genotypes which cannot survive under these circumstances. All right, let's do a little bit of mathematics. You started with how many moths? 2. Natural selection has eliminated 1. How many you got left? 1. All right. Has natural selection now made more or less? Less. That's right. All right. Natural selection has taken 2, eliminated 1, and you have 1 left. Now my question to you is the following. How can you use a process, natural selection, that makes less and less to make more and more? Because you start with few animals and now you want to get lots and lots of animals on the planet. Isn't that right? How do you do that? You cannot use a mechanism that makes less and less to make more and more, so this doesn't make any sense. In the past, there used to be a lot more variety than there is today, so natural selection is surely doing away with things. Now, here you have the dog family, and you have small dogs to large dogs, and you have all these dog shapes and all these ear shapes and all these strange little creatures. Look at the ear shapes. Long, droopy ears in the bloodhound, this ear in the collie, and all these hair types that we see over here, all of them present in the dog family. Question. Obviously, the ancestor, all these dogs were bred from the wolf, must have had genes for all of these appearances. Isn't that right? Must have been there. Question, where did the genes come from? 2 choices. Chance or design for all of that variety. It didn't come about by natural selection. Natural selection doesn't create anything. It only chooses between things that are already there. So in the past, they would say every single one of these snails is a different species. Today we know this is one species. Under some circumstances, some sets of genes are activated. Under other circumstances, other sets of genes are activated. And so, this is actually one set. Some butterflies, for example, have different appearances in different seasons. And some of these creatures, like this pigeon, actually must contain all the genes for what has been bred out of the wild pigeon. That means large pigeons, small pigeons, black pigeons, white pigeons, funny pigeons. All kinds of weird pigeons have been bred from the wild type. Where do all those genes come from that were there? Either they were built in by some designer, or they came into existence by boom, little house, boom, little house, boom, little house. That's what you have to believe in terms of the probability. Look at the variety here, from black to white, from black to white, from small to large. It's an interesting creature. It's known as the quagga. It's an extinct type of zebra that only had stripes up to a certain area, and then the rest was stripeless. And it went extinct some 300 years ago. And then they found a skin in a museum, and while they were cleaning it, the taxidermist discovered, wow, there are some blood vessels still stuck to the back in the dried fat. And that contained red blood corpuscles. So what did they do? Or blood cells in general? They sent it for DNA analysis. And when the